I try to deliver the material that should be delivered by Professor Kanaya, and also I will continue to the more detailed material about the protein-protein interaction and also drug target interactions using machine learning. Okay, maybe I will share my screen first. Already, can you see my slide, Miss Adila? Okay.
Slideshow.
Not yet?
Not yet.
Okay.
Oh,
is it
yes,
okay?
It is okay. Okay.
Okay. In this session, I would like to deliver material about the network pharmacology road on jamu modernizations. Firstly, I would like to introduce myself. My name is Wisnu Ananta Kusuma. I am now a lecturer in Department of Computer Science, IPB University, and also Executive Secretary in Tropical Biopharmaca Research Centers. Maybe I will just introduce first about my background. Actually, I come from Semarang. I was studying in SMA 3 high school in Semarang, and then I got a undergraduate on aeronautics in Bandung Institute of Technology, and also a master in informatics at Bandung Institute of Technology also. And then after graduate from ITB, I work at Indonesia Aerospace at astronautic division as a real-time software engineer. And after joining to the Department of Computer Science, IPB University, then I continue to pursue my PhD from Computer Science, from Department of Computer Science, Tokyo Institute of Technology, especially in bioinformatics lab. Okay, now I'm a lecturer in Department of Computer Science and also a researcher in Tropical Biopharmaca Research Center, and become the Vice Chairman of Indonesia Bioinformatics and Biodiversity Society from 2018 till 2020. So this is the brief description about my background. Okay, we start the material. As you know that Indonesia is very rich, the rich biodiversity in Indonesia, occupying only 1.3% of the the world's land surface. Indonesia is one of the world's richest nation in terms of its biodiversity. Maybe the most richest is Brazil, but Brazil actually the biodiversity mostly located in Amazon. But if we look at Indonesia, the biodiversity actually spread of many locations in Indonesia. So this is become the advantage for Indonesia, I think. Indonesia is an archipelago made up of about 17,000 islands, throughout which there are unique ecosystems containing a large number of diverse species. And also it possesses 10% of the world's flowering species, about 25,000 flowering plants, and ranks as one of the world's centers of agro-biodiversity of plant cultivars and domesticated livestock. So this is the rich biodiversity in Indonesia. But there is a gap from the paper written by El Fahmi. The area of Indonesia tropical forest covers about 143 million hectares and is home to about 80% of the world's medicinal plants. And the Indonesian country study on biodiversity estimate the number of flowering plants. I already mentioned before that consists of 25,000 until 30,000. And also PTAC published the dictionary of Indonesian medicinal herbs containing more than 2,500 plant species, which principally can be developed for medicinal purposes. And also Professor Suhut from IBB University identified that there are 1,845 species with medicinal potential in the forests of Indonesia. So this is the big potentiality. However, from the BPOM is like a FDA in America, from BPOM report, we know that only 24 products of phytopharmaca can deliver to the society for commercials. And also only 62 products as a standardized herbal medicine. So it's very, the gap is very big, yeah? Because we have, we are the biggest biodiversity nation, but only can utilize the biodiversity, become very few phytopharmaca and standardized herbal medicines. So this is the problem that should be solved and should be think by all of us as a researcher or academia, and also industry maybe. And there is a motivation, I think, that so we maybe all of us already known about the TCM, traditional Chinese medicine, and we know that traditional Chinese medicine also be prescribed or be also be used by a doctor in Tiongkok to cure patients for many diseases. Why the traditional medicine can be developed, can be growing, still growing, and become more more popular, because traditional medicine actually applied the system biology. There is a paper published in 2007 in Nature, state that if there is any technology that could lead to a breakthrough in traditional medicine, it will be system biology. So researchers in China already use the system biology for developing the traditional Chinese medicines. So the question is, how about jamu? How about jamu? Dr. Danang already explained about scientification of jamu. So this is our homework, how we can also adopt the system biology in our cases in developing jamu, herbal medicine, traditional Indonesian traditional herbal medicines. Well, before we talk about it more detailed, I would like to just give you a overview about system biology. Actually, there is two approach here: reductionist approach, which is based on the genome sequencing, the DNA arrays and proteomics become the basic information, then to make analysis of something, for example, for biological process and so on. And also there is an integrative approach, which is use bioinformatics, also system biology and modeling or simulation based analysis. From reduction approach, actually they work in molecular level, such as gene, protein, structure, and function. And function is studied at the molecular level. and then the integrative approach will bring this molecular level molecular level analysis result to system level. So actually, this is the important role of integrative approach, which is including bioinformatics, maybe metabolomics, and so on, to bring the information from gene, proteins, other omics, become the new knowledge in the system level. The representation of system level here is interaction of components in the biological system. So we do not only see the interaction between one, for example, one compound to one target, one protein target, but maybe in this perspective, we try to move from the one compound, single compound, single component, single target into multi-component, multi-target. And many papers already highlight this, the importance of the system biology. For example, drug discovery in the age of system biology. There are research umbrella here that drug target interaction is very important in this era. Because using the system biology, this tend to make improvement. For example, this is because the rise of chemical approach for data integration. So we not only use one or single data, but we use many types of data. Maybe Dr. Sony already explained about multi-omics. So many data from multi-omics will be integrated to analyze what actually is there any pattern, what actually important in this integration of data. This paper that mentioned about how the traditional medicine, that pharmacology, use this paradigm to use the system biology for improving their approach. For example, in this diagram, sorry, I will just use the pointer. Yes. There is a help network. The help network actually draw the interaction between compound and compound, for example, and also target network. There is actually content of gene or proteins, and also disease interaction. For example, if we look at the COVID disease, then we can say that there is a comorbid disease also appear in the COVID patients. Okay, so how we combine or integrate these three type of network? This is actually how traditional medicines use system biology to improve their approach. So now currently, sorry, this is. Did you still hear my voice? And what actually in this message, low system resource may affect? Sorry. Okay. So currently we know about the the term of we call it as network pharmacology. Yes, network pharmacology actually is application of principle of network theory. So we also have the discipline in science, we call it as mathematics. So we already know, all of us learn about mathematics. And there is a subject in mathematics we call it as the graph theory or network theory. That's very important for analyze the system biology, to apply or to utilize system biology to complex dynamic organic interconnection and molecular system, because our body actually consists of many cells, many components like protein, maybe enzyme, hormone, and so on, that they are not work by themselves, but they conduct the interaction, the complex interaction. So we cannot separate this. If we try to analyze the mechanism, we cannot separate into component per component. I said we cannot because actually what the condition of some, for example, one component will be influence will influence the other component. For example, if there is no balancing in our metabolism, then we will get the sick, for example, like that. So because the system biology is very complex, so we try to use the tools that can make us simple, so that we can so that the interaction is not so complex. Maybe there is many assumptions that we so take, but we can make the complex system become more simple by using graph theory, because we can model, for example, protein or gene or disease as a node, and then interaction as a edge or link. After we got the graph, then we can apply the graph algorithms or graph theory, the concept or principle in graph theory, to analyze the graph. The aim or the objective is to find the interesting pattern, that represented the characteristic of interactions. There is the important part that should be understood by us. I already mentioned before about the one compound, one target, and multiple compound, multi-target. In this slide, we saw the comparison between ordinary medicines, drug that already delivered, and maybe all of us use this drug to cure disease, for example, drug that already approved by FDA. We can also say that it's like a synthetic drug, for example. This actually the synthetic drug or modern drugs or ordinary medicines use the paradigm of one compound, one target interactions. So one compound, one compound will target the, for example, protein, a certain protein. You will get this lecture from tomorrow from Dr. Satyanto Triwahyudi. Yes, like this. So if, for example, compound can have the have, for example, the low energy affinity or have a good interaction, then we can say that the efficacy of this drug is good, for example, like that. But in the paradigm of multi-target and multi-components, we not only focus on the single component, but we try to consider many components that actually contains in herbal, for example, medicine. In this case, in the slide, we saw Campo medicine from Japan, and maybe we can change this like jamu from Indonesia. So the mode of action is multi-component, multi-target. So we cannot analyze this like maybe like a single component, single target. There is one that is the difference approach, I think. And some of you already asked in the Pak Sony session that what about the synergistic of among components, for example, because if we drink jamu, yes, for example, jamu kurkuma or jahe itu apa ya? Singaber officinale, sorry. Actually, there are many compounds inside the jahe or the Singaber officinale. So what this is only for from one plant. Usually jamu consists of many several plants. Yeah. For example, jahe or Singaber officinale, then curcuma and so on. So if we drink this jamu, then actually there are many compounds that will make an interaction, and so calculate the synergetics. So the synergetics effect is become an important issue, because in medicine there are two important issues. The first is efficacy, and the second is safety. So we curious if for example 2 or 3 or more compounds consumed together, then will make for example increase the toxicity and so on. So this become the important issue. But in this lecture, I would like to show you how we focus on how to use the network theory here, or graph theory, to represent or to analyze the mechanism or system biology related to complex dynamic organic interconnection in, for example, in regulating in certain disease, for example. And I didn't want to go detail about how the biological process or chemical process and so on, because I'm from computer science, so I cannot explain about that. Okay. This is actually already mentioned before that combinatorial effects can make synergetic effects or antagonistic effect. So the screening on purified component or using single target prohibits the discovery of the most important synergetic nature of natural products. Okay, this is the multi-dimensional pharmacology of complex mixture. I cannot explain more detail because it's not my expertise. I just want this slide. I just want to use this slide for show you that there is actually combinatorial effects. For example, if we combine some component, can make increase synergetic effects or decrease or maybe antagonistic. So actually the aims or the objective of summer course, the summer course is try to socialize our new, we call it as new paradigm. So actually the researchers from tropical biopharmaca research centers, including Mr. Rudy Herianto, Dr. Fari, Dr. Muhammad Rafi, and also Professor Imanida, Dr. Farid Afendians, and many researchers try to introduce the new paradigm. We hope that this paradigm will be discussed widely. Yes, widely in pharmacology, maybe chemistry, chemistry society, or biochemistry society, or biology society, pharmacology society, and maybe from also medical medical science society, for example, like that. So we arrange this summer course. We try to introduce the maybe it's not a real all of in this program is real all of is new, but actually we try to read papers and to join the conference and so on, and then we try to summarize or to formulate to develop the framework. That there is five important point if we want to improve or to use the multi-drug, multi-target paradigm to develop the multi-target, and multi-drug paradigm. The first point is related to in silico and bioinformatics approach. So we if, for example, to develop the traditional medicine, usually researchers come from the information that passed down generation to generation. For example, I would like to, for example, many researchers maybe have a privilege of subjective choice for do research in specific plant. For example, I just want focus to make a research about curcuma or about jahe, for example, like that. But in our perspective, because we know that there is a gap between utilization of biodiversity, so we try to introduce the new thinking. I mean that why how if we start to explore the biodiversity data, especially for example plant and so on. So we do not start from the specific plant, but we explore, we collect a lot of plants, then using the in silico and bioinformatics approach, especially machine learning and so on, to explore which actually and screen which actually the candidates, the potential candidate plant for become jamu formula for specific disease. So we don't start from the specific plant, but we collect a lot of data because this is a big data era and also the era of artificial intelligence. So we try to use both of them, the power of the big data and artificial intelligence, to explore many data and then try to screen to reduce this research search space, to reduce the search space, or to reduce the combinatorial problem, yeah, to get the potential candidate of compound or plants. This is maybe I hope we understand this perspective. And then after we got, for example, some candidate, we will do the common procedure in drug discovery, for example, toxicity, efficacy, but maybe we should think about how to improve the procedure that actually apply to the one component, one target, so that they will be suitable for multi-drug, multi-target. For example, I have experienced that when we got some component, some candidates plan that that have a potentiality to be compound that can prevent COVID disease, can target, can have a target from the, for example, main protease of COVID-19 from the Pisidium guajava, yes, jambu biji merah. There is a concern that BPPOM asks us, yeah. I mean, the procedure of BPPOM state that we should conduct the pharmacodynamics research, pharmacodynamics experiment work in wet lab. So it's very difficult because pharmacodynamics need the virus, yes, the virus. So we think why we cannot just bypass this step, so that after get the candidate result from in silico and machine learning approach, we can do clinical trial. But BPPOM still state that it's not can be, we cannot do that. We should strict with the procedure that the firstly is conduct the pharmacodynamics work. I'm not pharmacist or chemist, so I cannot explain more detail about that. But the question is, if, for example, many pharmacists and chemists sit together to discuss this problem, is there any procedure that more suitable for multi-drug, multi-target? For example, there is improvement in this second step. For example, after get a candidate from in silico, then maybe we can bypass, for example, pharmacodynamic or something, that because the interaction and also the mechanism of interaction between component among components or component and target already explained in the in silico analysis, drug target interaction analysis, for example, like that. And the second step is about the standardization of material, because, for example, plants grow in different location will have different metabolite, for example, like that. And we still follow the procedure to conduct the preclinical and also clinical, preclinical and clinical trial, and then also try to use the modern approach like metabolomics here to ensure what can actually the metabolite, the active metabolite that important in this mechanism of curing or preventing something, some disease, for example, like that. So this is actually the step, the important point for developing multi-drug, multi-target. And then we propose the IJA analytics workflow. IJA analytics workflow, and then this workflow actually will be implemented or applied in IJA analytics application that developed by Tropical Biopharmacology Research Center. The first thing is find significant proteins by reducing this network into more few subnetwork. From protein-protein interaction network, the big protein-protein interaction network, we try to reduce the dimension of this network, become the small, so that the protein that contain in small network, we assume that that protein is very important related to the mechanism or metabolism or regulation of related to certain disease. We already conduct many research like based on topology, skyline query, clustering, and so on. And the second step from IJA analytics framework is predicting link interaction of compound protein network based on machine learning techniques. This, if you hear the material from Professor Yaya Rukayadi, Professor Yaya said that the most challenge in the JAMU develop modernization JAMU is because we don't have many information. We don't have information about interactions between compound contains in herbal or JAMU or plants with the protein target. There is still there is no database that contain comprehensive data that show the interaction between herbal compound and protein target for specific disease. So here we try to solve this problem by introducing the method by using the machine learning and other methods for predicting link, lack of link or lack of interaction of compound protein. We call it as link prediction using BLMNE, deep learning, and also we try to, because to predict this link, we need information of interaction between commercial drug, commonly drug that used by society, for example, synthetic drug or drug delivered already proved by FDA, and the target interaction, because many database, for example, in drug bank or super target and so on, contains of this information, drug or from the compound from the drug registered drug with the protein target. So to solve or to find the interaction between herbal compound and protein target, we should measure the similarity of compound in herbal to compound in registered drug. If the similarity is high, so we can assume that compound in herbal have interaction with protein target as compound registered compound as because the registered compound have the interaction with protein target. And after we got the interaction between herbal compound and protein target, then we can performing to find the JAMU formula by using graph traversing. Yes, this is the graph traversing here. Yeah. So we can, for example, do from disease, specific disease. For example, I would like to have a formula for diabetes mellitus. So we just doing the traversing from disease to plant. Or if we already have a formula, you can validate this formula to know what kind disease that can be targeted by this formula of plant or jamu formula. So we can find the formula from the plant or from the disease. After that, we should calculate the synergetics effect between, for example, many compounds. But in this case, we still only can calculate the synergetic between two compounds. We still not can calculate the for example 3 or 5 compounds like that. This is open research. Okay. So the first step, find the significant protein from protein-protein interaction. The objective is how to reduce this network become the small network that consists of important protein or significant proteins related to specific disease. Many approaches can be proposed. For example, we use the topological approach. And the step is find subnetwork based on shortest path. Shortest path is, for example, if you, for example, A, B, C, there is like that. So the shortest path from A to C is not A, B, C, but from A to C, like that. Okay. Then, sorry, maybe I will continue because I have two material. So I will continue my material without break. Yeah. Okay. Is it okay, Ms. Sadila? Hello?
Maybe we have a break.
No. I will continue my material. So is it possible if without break?
Oh, okay. Okay, huh? It's okay.
Yes, because I think this is related material. This material is related each other. So I think it's
okay.
Better if I continue the two session, and then maybe there is no break in this session.
So you will deliver PPI analysis?
Yes, after we'll continue before break. Without break. Okay.
Yeah.
Yes.
Okay.
So thank you. Okay, I will continue this explanation. First is find subnetwork based on shortest path. This is actually shortest path, and then calculating the topology metric connectivity, for example, degree between the centrality and closeness centrality, and then find significant protein based sorting protein based on the topology metrics. It's already published in the paper in computer engineering and application. I will show you the explanation of degree between the centrality and closeness centrality in the next material when we talk about the protein-protein interactions. But I just want to show you that between the centrality actually is a node of protein that have a role as a bridge. Yeah. So if this protein is deleted or omitted, then the network will divide into two or more network, more than one small network. Okay, or this network will divide into sub network. This is between the centrality, and degree is actually, for example, this node is connected to 1, 2, 3, 4, 5, 6. So degree is the number of connection of one node to another node. So this node have 6 degree, for example, like that. and shortest path I already explained, and the closeness centrality actually is how the metrics that measure how the one node close to another node, how close one node to another node, like that. So in the experiment, we build the protein-protein interaction of diabetes mellitus. We got 83 protein candidates and have a 2053 protein interaction. And when we draw the network is like that, but using the topology-based approach, we can reduce the network, become the small network like this, that consists only 24 significant protein candidates and 674 protein interactions. So from the 24 protein significant candidate, we identify, for example, insulin, AKT1, I don't know what actually this protein or gene, insulin receptor and so on, but there is a description here. Sorry, I forgot to make a translation that insulin is reduce the concentration of glucose, glucose blood. Yeah, and I mean I think you from the chemist or biologist already know about the function of this protein. I just want to show you that from this approach we can identify the important protein that have an important role actually in, for example, in diabetes mellitus. And we also can use the other approach adapted from maximum point problem in math. So if you studying math, that mathematics, you will find the you will meet the maximum point problem. Actually, this is adapted to be a scaling query algorithm. This dominant scaling can find scaling point. We define the scaling point, which is more important and meaningful in the high dimension space. This actually analogy with, for example, if you have a child or a brother still in elementary schools. In my era, only one child or one student that become a champion in one class. But now the modern education changed the perspective. They don't want, they didn't do like the old era that only have and that only give an award for the best students. Yeah, only one best students, yeah, in the class. But now change to give award for many students. For example, for students that have a biggest score in math, they give award to the students. So there is many champion, many best student here. Best student for mathematics, for example, best student for science, best student for sport, best student for sing and so on, art and so on. So the concept of this best student in elementary school actually analogic with the scaling point. So the scaling query is we will find or screen the point that sorry committees, please move the so the objective of scaling query is to find the points that minimum can dominate the others point. Minimum can dominate the others point, like the best students in elementary schools. So by using the scaling query, then we find a protein, also a gene that related to diabetes, like insulin, AKT1, STAT3, and so on. And when we compare between using shortest path and scalene query, there is some protein that overlap here. So this is interesting because we use the different approach. Then the result is different, but still have three protein or gene that overlap. Maybe this is the maybe I don't know. I am not expert in diabetes, so maybe we can analyze. So in the final project, your problem is like that. Try to find the protein that have an important role in this related to the specific disease. And I hope, because your background is in from the non-computer science, maybe from pharmacy or chemist and so on, so you can make analyze more deeply about your findings about the protein, the important proteins from protein interaction. This already published in international conference, has got a best paper award, my student. And the result, sorry, this is diabetes, and this is we try to implement from the to Parkinson disease. And this is the result. Protein included in top 21 scalene query are SNCA, PARC2, and so on. And significant protein for Parkinson disease based on this study is the most significant protein is SNCA, or the most important protein is NCA, that has been proven to have significant role in Parkinson disease. We only conduct the literature study, but this is open open collaboration. Yeah, if maybe we can provide the important information about the important protein from for specific disease, and then maybe the chemists or biologists or pharmacists can continue to do research by using wet lab to maybe to find the biomarker also. And the second step in IJA workflow is link prediction. The problem is many herbal compounds do not have link to proteins, so the we should answer, is there any connection between this compound and protein, like this, yeah, between this compound and protein. So we require link prediction, yeah. And to conduct this, we should calculate the similarity of compound and the similarity of proteins. So for example, we try to calculate the similarity score between this compound and also this compound and with all compound here, and also calculate the similarity of proteins. Similarities of compound was calculated using fingerprint algorithm that is already described by Dr. Sony, and the similarity of protein was calculated using, for example, many approaches. So, for example, using the sequence alignment, using Smith-Waterman algorithm, or maybe just using dipeptide composition and so on. So, if, for example, this similarity of compound 1 and compound 2 is very high. This is compound 1 and compound 3 is very high. So because compound 3 has interconnection, has connection with protein 2, for example, protein 1 and protein 2. So we can conclude that compound 1 will have a interaction with protein 2, for example, like that. So we calculate all the combination of compound and protein by using bipartite local model, network interaction profile, and frame. So actually the principle of a bipartite local model is using similarity between compound and protein to make a decision whether the compounds have a interaction with this protein. And after we have many interaction between compound and protein, then we construct the network, we expand the network not only from protein and compound, but also we connect to the plant. And if we, because this is specific disease, if we make this model general, so we also can connect to the disease. We get information of plant from knapsacks or Jamu IBB, and for protein from Uniport, BioAssay, and we got data of disease and protein from OMIM, and also compound from PubChem and Drug Bank and so on. After we, because we would like to know if whether compound and herbal have a interaction with the protein or not. So we need the information from compound that we collect from delivered or registered drug or conventional drug. This is maybe the brief description about the method. First, we connect graph from between plant, compound, and protein, and then we connecting graph A and B. Actually, A and B have already connected, but B is extension of A because in B we contain a compound from herbal, but in A is contain a compound from conventional drug. Then we try to calculate the similarity between S2 and S3, and also mapping the protein that targeted by compound. For example, P1 and P1 here is equal, so the similarity value is 1. And then we also using fingerprint technique to calculate the similarity between compound in network A and compound in network C. Compound in network A is compound that we collect from the drug or conventional drug, and the compound in network C is from the herbal. Then after that, we, after doing the repeating the procedure, then we store the information here. So each compound and will be drawn or will be stored with their pair, protein pair. For example, S1 accent, X1 from herbal have interaction with P1 and so on, and also store the value of similarity value. And we traversing from protein into compound until to plant, and also store the information. For example, if we got the higher value, then the information of value of similarity value is will be updated with the highest. Yes, like that. And after that, we will get this information between compound and the connection with protein, with the information of the similarity value, the accumulation of the simulation of similarity value, and also we store the plant and their connection with protein, and also store the information of similarity value. And then we accumulate this score to get the overall score for each plant, so that we will get, for example, T1 will get higher score, and T is, for example, lower score. We like this. Then we can rank which plants actually have a higher score related to specific disease. In this case, for one plant, this is very surprising that it is based on our data. The Magnifera indica have a highest score formula related to diabetes mellitus, and also maybe bunches and so on. And we already try to know or to identify what part of the Magnifera indica that maybe have a compound that can be used for diabetes mellitus. Actually, is from the seed. And if we combine two plants, there is also information, interesting information: Citrus aurantium and Magnifica indica. So until this slide, we can see that if usually researcher conducting the research about the specific plant, for example, that have a potential to cure diabetes mellitus, but in our case, we don't have information about the plant that have important role in the diabetes mellitus. But by using the network analysis, we can propose a new plant, maybe new findings. Citrus aurantium and Magnifera indica maybe can have potential to cure or to prevent the diabetes mellitus. But then this is it's not my duty or my role, but this is maybe should be continued by researchers from chemistry or biochemistry or medicine or something. And after that, we try to use the to calculate synergy score. Yes, we collect the interaction protein from string DB, and also we need information of phenotype data related to the specific disease from the OMIM database and from human phenotype ontology. And this score, synergy score, actually can be calculated from topology score that related to PPI network, and from agent score that actually related to phenotype related phenotype of the disease. For topology score, I will not explain detail actually because there is a specific material and protein interaction. Yes, but actually there is the three metric that we use here to calculate this formula. And also we use the phenotype ontology HPO to show to calculate the distance between the, for example, the phenotype that targeted by specific compound with other phenotype that targeted with other compound. For example, we have two compound. First compound actually is related to abnormality of the hips, and the second compound actually related to congenital hip dislocation. Then the distance is 2. This is the simple explanation like that. Then we calculate the Akin score with this formula, and then by multiplying the Akin score and topological score, we will get the synergistic score. For example, like this compound sulfurfluorine and this fluorine have the highest synergy score, and this is the pair of compound related to the diabetes. And we try to use to implement, but not all features already implemented. You can access IJAH. For version 2 is IJAH APPS CS IPB AC ID. For version 3 is IJAH Biofarmaka IPB AC ID. In this version 3, we already have a synergistic calculation. Now, we have also SNP. SNP is application for identification and identification of single nucleotide polymorphism, and also calculate the genotype phenotype association. If we can, for example, know the SNP here, and we can make association with the phenotype. For example, Delta virus of SARS-CoV-2 have a SNP in this, for example, in the RNA DNA, in the DNA, and then we can calculate the association with why the Delta is more spreading, easy to spread to the other people, for example, like that. Or about the diabetes mellitus and so on. And this is actually the future work, our dreams that we would like to. We already have IJAH and SNP, and now we are working to develop I prime, ABB precision herbal medicine discovery. So we combine the information of genotype from genomics space with the information from the maybe it's from the drug target space, so that we combine this, become the IBB precision herbal medicine or precision herbal medicine. That so we hope that we can produce find the potential herbal compound that can occur the specific disease effectively, or have a good efficacy, but also safety. Try to minimize the, for example, toxicity or adverse reaction drug, like this. Thank you. This is the most the first material. Before I continue to the second material about protein-protein interaction, is there any question maybe, or this moderator is also Mr. Ahmed? Mr. Ahmed, we will continue the other material, or just break for question answer first?
Probably we could give 3 minutes if we have questions. Maybe we can answer the questions. Or if we don't have questions, maybe we can continue to the next lecture. Then we will have maybe questions from this lecture and the previous one. Meanwhile, while we are waiting for the questions from this lecture, we just want to say thank you very much for stepping up. We know that this lecture wasn't yours to present in the first place, but due to the absence of the original presenter, you stepped up, and that is very, very brave, and it shows some level of leadership. So we want to appreciate you for that. And I think this lecture has showed us not just, it has given us not just information, like passive information, but also active information in like giving us multiple resources, websites, and tools that we can use in working, most especially in network pharmacology for traditional medicine. And it has also given us roadmaps, pathways, and how we can go about to do this work. So as we are waiting for our questions, maybe we have exactly 1 minute, and maybe by 10:51 or 52, we can continue to our next lecture. I believe by then our listeners, our participants will have their questions properly drafted, and they'll have a complete idea of what are some of the questions they would like to know, and what are some of the difficulties or lack of clarity they have gotten from the lectures that they will want to clarify. But so far, I've seen an interesting lecture here. I've seen a potential research field where students can venture in and do a lot of research, a lot of resources, and a lot of opportunities are right here. Since we don't have any questions till now, I think Dr. Kusuma can continue to the next lecture, and then we can go for our break, our long break.
Okay.
Thank you.
Okay. Thank you. Yes, I will continue with the second material, actually related to protein in the. Sorry, I will share my screen.